For most of the history of genetics, DNA mutation was treated as a purely classical mechanical process, one governed entirely by ordinary textbook chemistry rather than anything stranger. A copying error here, a chemical reaction there, radiation damage, replication mistakes, all explained using ordinary chemistry and ordinary physics, the same kind of cause and effect processes you'd use to explain why metal rusts or why a photocopier occasionally smudges a page. Nothing about it seemed to require anything as strange or as counterintuitive as quantum mechanics. According to a growing body of research over the past 2 decades, that assumption was incomplete, and one of the strangest branches of physics turns out to play a genuine measurable role in how genetic mutations actually occur at the molecular level, deep inside the double helix of DNA, at a scale far smaller than anything visible under even the most powerful conventional microscope. Researchers have found evidence that a distinctly quantum phenomenon called quantum tunneling may be quietly influencing the rate at which certain mutations appear, a finding that connects the strange rules governing subatomic particles directly to the deep evolutionary history preserved inside ancient human genomes, and to nearly every genetic study built on top of that history. To understand why this matters, and why it took physicists rather than geneticists to first notice it, it helps to walk through exactly what quantum tunneling actually is, since it's a genuinely strange concept even by the standards of modern physics, one that challenges basic intuitions about how solid physical matter is supposed to behave. In classical physics, the physics of everyday human-scale objects, if you don't have enough energy to get over a barrier, you simply don't get over it. A ball rolled toward a hill will roll back down again unless it has enough energy to reach the top, and continue over the other side. Quantum mechanics, which governs the behavior of particles at the subatomic scale, breaks this rule in a way that still unsettles physicists who've spent entire careers working with it. Subatomic particles, including protons, don't behave like solid little balls with a fixed, definite position. They behave more like spread-out probability waves, existing in a kind of blurred, uncertain state until measured. One consequence of this strange probabilistic behavior is that a particle can sometimes appear on the other side of an energy barrier without ever actually possessing enough energy to climb over it in the conventional sense, essentially tunneling directly through the barrier instead of going over it. This isn't a hypothetical curiosity confined to abstract physics experiments. It's a well-established, extensively verified phenomenon, and it plays a critical, practical role in several areas of chemistry and biology, including, as it turns out, inside the DNA molecule itself. If you find it fascinating that something as strange as particles briefly existing in multiple places at once could have any bearing whatsoever on biology, that's exactly the kind of connection this research is built on. Go ahead and hit that like button, because the specific mechanism involved here connects quantum physics to genetics in a way most people have never encountered before. The connection to DNA comes down to hydrogen bonds, the relatively weak chemical bonds holding the 2 strands of the DNA, double helix together, formed between specific pairs of bases, adenine with thymine, and cytosine with guanine. These hydrogen bonds work because a hydrogen atom, essentially a single proton, sits in a particular position, linking the 2 bases together. Under normal circumstances, this proton sits in one of 2 possible positions, corresponding to the standard, correctly paired arrangement of DNA bases that gets faithfully copied every time a cell divides. Here's where quantum tunneling enters the picture directly. Research building on theoretical work first proposed decades ago, and considerably refined more recently using detailed quantum mechanical modeling, suggests that the hydrogen atom involved in these bonds can occasionally tunnel from its normal position into a different alternate position, purely as a result of quantum mechanical uncertainty, rather than any conventional chemical trigger like radiation damage or copying error introduced by cellular machinery. When this tunneling event happens at exactly the wrong moment, specifically during DNA replication, it can cause the cellular machinery responsible for copying DNA to briefly misread which base is supposed to pair with which, introducing what's called a tautomeric mismatch, a subtle temporary alteration in the hydrogen bonding pattern that can result in a permanent mutation if it isn't caught and corrected by the cell's proofreading machinery before replication finishes. This means that at least some fraction of DNA mutations, the same mutations responsible for driving evolutionary change across the entire history of life on Earth, may originate not from any external damage or conventional chemical error at all, but from an underlying quantum mechanical property of the hydrogen atoms holding DNA's base pairs together, a property that exists regardless of environmental conditions, background radiation, or replication errors introduced by cellular enzymes. If uncovering how deeply strange subatomic scale physics quietly shapes something as fundamental as genetic mutation fascinates you, make sure you're subscribed, because this exact kind of connection between physics and biology keeps surfacing in genuinely unexpected corners of modern science. The idea itself actually traces back further than most people would expect, originating with a Swedish physicist named Per Olof Lodin, who first proposed the concept of proton tunneling within DNA base pairs all the way back in 1963, just a decade after Watson and Crick had published their landmark structure of the double helix. Lodin's original proposal was remarkably prescient for its time, suggesting that the same quantum tunneling effects known to occur in simpler chemical systems could plausibly occur within the hydrogen bonds holding DNA's base pairs together, potentially contributing to spontaneous mutation independent of any external damage. For decades, this idea remained largely a theoretical curiosity, difficult to test rigorously given the computational limitations of the era, and it existed mostly as a footnote within specialized biophysics literature rather than as an active area of mainstream genetic research. It's only within the past 15 to 20 years, as computational quantum chemistry techniques have advanced dramatically and become powerful enough to model these effects with genuine precision at the molecular scale, that researchers have been able to meaningfully test and refine Lodin's original hypothesis using tools he never had access to. This research remained largely theoretical for a long time, proposed mathematically and explored through quantum chemical modeling long before researchers had the tools needed to directly observe or test it at the resolution required, given how technically demanding it is to model quantum behavior occurring within a molecule as large and complex as DNA. More recent work, using considerably more advanced computational quantum chemistry techniques than were available when the idea was first proposed, has strengthened the case that this tunneling process occurs at a rate genuinely relevant to biological mutation rates, rather than being so rare as to be effectively negligible. Some of this modeling work suggests that proton tunneling could account for a meaningful, though still relatively modest, fraction of the spontaneous mutations that occur during normal DNA replication, alongside the far better understood conventional sources of mutation like chemical damage, radiation exposure, and replication errors introduced by imperfect enzyme activity. Testing this idea experimentally, even indirectly, has required considerable ingenuity, since directly watching a single proton tunnel between two positions inside a living cell remains far beyond what any current laboratory technique can actually observe in real time, given the sheer speed and scale at which these quantum events would need to be captured. Instead, researchers have relied on a combination of approaches, including detailed quantum mechanical simulations that calculate the probability of tunneling occurring under realistic biological conditions, spectroscopic studies examining how DNA base pairs behave at extremely low temperatures, where quantum effects become easier to isolate and measure, and comparative studies examining mutation patterns across different DNA sequences to look for signatures consistent with tunneling-driven mutation rather than purely chemical or environmental causes. Each of these approaches comes with its own limitations. Low-temperature spectroscopic studies, for instance, can clearly demonstrate that tunneling occurs under laboratory conditions, but translating those findings into an accurate estimate of how often it happens inside a living cell at normal body temperature requires additional theoretical modeling, since temperature substantially affects the rate at which quantum tunneling occurs. Despite these challenges, the convergence of multiple independent lines of evidence, theoretical modeling, low temperature, experimental verification, and comparative genetic analysis has steadily strengthened the case that this isn't merely a mathematical curiosity confined to abstract physics papers, but a genuine, measurable contributor to biological mutation. It's important to be careful and precise about exactly how significant this contribution actually is, because this remains a genuinely active area of scientific investigation, rather than a fully settled, universally agreed upon conclusion. Quantifying exactly what percentage of naturally occurring mutations originate specifically from quantum tunneling, as opposed to more conventional chemical and environmental sources, requires extraordinarily precise theoretical modeling, since directly observing a single tunneling event occurring inside a living cell in real time remains far beyond current experimental capability. Different research groups working on this question have arrived at somewhat different estimates depending on the specific theoretical assumptions and computational methods used, and the field continues actively refining these models as computational chemistry techniques keep improving. There is an additional layer to this story worth exploring, involving how cells actually manage to keep this quantum instability from causing far more genetic damage than it actually does. Living cells possess an elaborate system of DNA repair enzymes, molecular machinery specifically dedicated to scanning newly replicated DNA for exactly this kind of mismatch and correcting it before it becomes a permanent, heritable mutation. Researchers studying the tunneling hypothesis have pointed out that this repair machinery likely acts as a kind of biological buffer against quantum-driven errors, catching and correcting the overwhelming majority of tautomeric mismatches before they ever become permanent, meaning that mutations that do eventually make it into the historical genetic record likely represent a small, imperfectly filtered fraction of the total tunneling events actually occurring inside cells. This adds an important nuance to the whole picture. Quantum tunneling may generate far more raw mismatches at the molecular level than actually surface as observable mutations, since the cell's proofreading and repair systems are actively working, constantly and mostly successfully, to catch and fix these quantum-driven errors before replication is complete. Understanding the true underlying tunneling rate therefore requires accounting not just for the physics of the hydrogen bond itself, but for the efficiency of this separate, entirely conventional biological repair system operating alongside it, a genuinely complex interplay between quantum physics and traditional molecular biology that researchers are still working to fully untangle. What can be said with confidence is that the underlying phenomenon itself, hydrogen bond proton tunneling occurring within DNA's base pairs, is grounded in extremely well-established quantum mechanical principles, the kind that have been thoroughly tested and confirmed across numerous other areas of chemistry and physics for decades. The genuinely novel and still actively researched question isn't whether this kind of tunneling can happen inside DNA at all, but rather how large a role it actually plays relative to other, better understood mutation sources, and how consistently that role has applied across the vast stretches of evolutionary time, separating ancient genomes from modern ones. This connects in a genuinely interesting way to the study of ancient DNA specifically, since understanding the true underlying rate and mechanism of mutation is fundamental to nearly everything researchers do when analyzing genetic material recovered from ancient remains, whether they're comparing Neanderthal genomes, tracing the deep ancestry of isolated populations, or reconstructing the spread of ancient pathogens. Techniques like the molecular clock, which researchers rely on constantly to estimate when two populations diverged from a common ancestor, or when a specific ancient lineage first emerged, depend entirely on having an accurate, well-calibrated understanding of how quickly mutations accumulate over time. If a meaningful fraction of these mutations originates from a fundamentally quantum mechanical process, operating according to its own independent probabilistic rules, rather than purely from environmental or chemical factors, this has genuine implications for how confidently researchers can calibrate mutation rates when interpreting ancient genetic material, particularly across the kind of vast evolutionary timescales involved in tracing human ancestry back tens or hundreds of thousands of years. There's a further connection worth drawing out here, specifically relevant to ancient DNA research rather than mutation in living organisms generally, since ancient genetic material behaves quite differently from DNA inside a living, actively repairing cell. Once an organism dies, its cellular repair machinery stops functioning entirely, meaning any chemical instability within the DNA molecule, including the kind of tautomeric shifts associated with proton tunneling, can no longer be caught and corrected the way it would be during an organism's lifetime. Ancient DNA, recovered from bones and teeth thousands or tens of thousands of years old, is already known to be heavily damaged through entirely conventional chemical processes, including deamination reactions that chemically alter individual bases over long periods of time. Some researchers studying ancient DNA degradation have begun considering whether quantum tunneling effects might contribute an additional, subtle layer to this post-mortem chemical instability, alongside the far better characterized conventional damage processes that dominate current ancient DNA research. This remains a considerably more speculative extension of the core tunneling hypothesis, and it hasn't yet been rigorously tested with anywhere near the same level of confidence as the basic tunneling mechanism itself. It does, however, illustrate how a piece of fundamental quantum physics research, originally developed to explain living cell mutation, might eventually connect to the specific technical challenges researchers face when trying to extract clean, reliable genetic information from degraded ancient samples. This doesn't mean existing ancient DNA research is somehow fundamentally wrong or unreliable. Molecular clock calibrations are typically built from extensive comparison against known historical events and independently dated fossil evidence, a practical, empirical approach that remains valid regardless of the precise underlying mechanism driving any individual mutation event. What this research does suggest is that our theoretical understanding of exactly why mutations happen at the rate they do remains genuinely incomplete without accounting for this quantum mechanical contribution, adding a deeper, more fundamental layer of explanation underneath the more practical, observationally grounded models researchers already rely on successfully today. This isn't the only place where quantum mechanics has turned out to play a genuine measurable role inside biological systems, and it's worth briefly mentioning that broader context, since it helps explain why researchers were willing to take an idea as seemingly strange as tunneling-driven mutation seriously in the first place. Quantum effects have also been documented in the way certain enzymes catalyze chemical reactions, using tunneling to speed up reactions that would otherwise proceed far too slowly to sustain life at normal biological temperatures. Photosynthesis, the process plants use to convert sunlight into usable chemical energy, has also been shown to rely on quantum coherence effects to shuttle energy through specialized proteins with remarkable efficiency. Even the way migratory birds sense the Earth's magnetic field appears to depend on a quantum mechanical process involving specially sensitive molecules in their eyes. Taken together, this growing body of research across genuinely unrelated biological systems has helped establish quantum biology as a legitimate, increasingly mainstream field of study rather than a fringe idea confined to speculative theoretical papers. DNA mutation via proton tunneling fits naturally into this broader pattern. One more example of life operating at its most fundamental molecular scale, according to the same strange quantum rules that govern the rest of the physical universe, rules that turn out to matter considerably more for biology than researchers assumed for most of the 20th century. There's something genuinely striking worth sitting with here, a reminder of just how deeply interconnected the different scales of scientific understanding actually are. The same strange, probabilistic quantum behavior responsible for phenomena like radioactive decay and the operation of modern quantum computers appears to reach all the way down into the molecular machinery underlying heredity itself, quietly influencing the raw material, random genetic mutation that evolution has worked with across the entire multi-billion-year history of life on Earth. Ancient human genomes, recovered from bones tens of thousands of years old and painstakingly sequenced using cutting-edge laboratory techniques, ultimately owe at least some of their genetic variation to a physical process operating according to the same strange rules governing subatomic particles, a connection that would have seemed like pure speculation not many decades ago, and one that continues to be actively researched and refined today. It's also worth pausing on why this particular story resists the kind of dramatic, reality-defying framing that sometimes gets attached to quantum physics and popular science coverage. Quantum tunneling in DNA isn't evidence of some mysterious, unexplainable force operating outside the known laws of physics. It's the opposite. It's a carefully documented example of well-established quantum mechanical principles, the same principles underlying technologies like MRI machines and semiconductor electronics, showing up in a biological context researchers hadn't fully appreciated until relatively recently. The genuine discovery here isn't that logic has somehow broken down. It's that the boundary between physics and biology turns out to be considerably blurrier and considerably more interesting than the traditional separation between these two. Fields ever suggested, with real, testable, incrementally refined science sitting on both sides of that boundary rather than any kind of unexplainable mystery. So the next time you hear about ancient DNA revealing some new insight into deep human history, it's worth remembering that the very mutations making that discovery possible in the first place may trace back, at least in part, to something as fundamentally strange as a single proton briefly existing in two places at once, quietly reshaping the genetic code, long before any of us ever developed the tools needed to notice it happening.